Hello, my name is James Shemkiewicz, and today I'm going to talk to you about the level of microbial contamination and assessment of shelf life in organic versus conventional spinach. So a bit of background information into organic versus conventional farming. So conventional farming makes use of synthetic compounds for fertilizing the soil, where this is forbidden in organic farming. So this natural method of farming requires more labor to ensure the contamination levels are kept to a minimum, and this also makes the produce more expensive. So the reasons why people may purchase organic foods over conventional is they're generally perceived as better quality, healthier, and contain more levels of nutrients. So this research project is the overall comparison between organic and conventional spinach at the post-harvest stage. So this is the stage where quality matters the most, as the consumers are the ones who are affected by the shelf life. So often consumers are having to throw out food due to spoilage of the product, where this has contributed largely to annual food waste production. So this is not only costly for the consumer, especially if this is done repeatedly, but it also takes a toll on the food waste industry. So there's evidence that suggests that even small increases in shelf life could result in a significant reduction in food waste at the household level. So food safety also plays a role in the minds of consumers, where E. coli is the main organism of concern. Although many strains of E. coli are not pathogenic, there have been reports of numerous foodborne outbreaks from the consumption of fresh spinach. So pathogenic E. coli strains, some produce the Shiga toxin, which can cause diarrhea, hemorrhagic colitis, hemolytic uremic syndrome, and other complications. So fresh leafy vegetables such as spinach are particularly susceptible to contamination of pathogens. So E&B agar was used to culture E. coli, where this was done in 1 to 10 serum dilutions up to the 10 to the 3 dilution. And these plates were incubated for 48 hours at 37 degrees Celsius for a total of 6 plates for E. coli. So Ammomedia Pseudomonas isolation agar was used for Pseudomonas, where a 10 to the 2.5 dilution was used because in the E. coli experiments there was a major decrease in the contamination levels between the 10 to the 2 and 10 to the 3 dilutions. And these plates were incubated for 24 hours at 37 degrees for a total of 18 plates. So this involved choosing 10 random leaves from the original spinach bag and then placing them into Ziploc bags. So this totaled 3 samples for each variety of spinach, totaling 6 samples overall. So the height of the spinach leaves were measured using a ruler in units of centimeters, and this was achieved by placing the leaf on a flat bench and measuring from the apex to the end of the petiole. So this was done individually for each leaf, and the average was taken from each sample. So both variables of height and weight will be measured at the beginning and end of the experiment. So the sensory assessment was actually not conducted in the lab, but given unrestricted laboratory access, it would have been done following the procedures set out by Medina et al. 2012, which assessed the spinach quality in terms of off odors, dehydration, and overall visual quality. So the limit of consumer acceptability is the rating at which determines the sample as not suitable for shelf life. So the end of the sample's shelf life will be when the limit of consumer acceptability is reached for all categories, and this was achieved by using a point scale system. So more than one type of colony was observed on the plates, where the larger mucoid-looking colonies were thought to be the species Klebsiella, and the smaller colonies were considered as presumptive colonies of E. coli because they didn't possess a green metallic sheen, which is known to be characteristic of E. coli colonies. Further biochemical tests would be needed for confirmation of the species, where the number of colonies could not be counted due to the number being outside the range of 30 to 300 colonies, which is used for statistical accuracy. So instead, observable differences in the overall contamination levels were compared between the two varieties, where the organic samples had more levels of contamination overall. So the results for Pseudomonas have produced a contrasting result to the E. coli experiment, where overall higher levels of contamination were seen in the conventional samples. This procedure was done in duplicates for most dilutions, and overall 18 samples were used for the experiment, compared to the E. coli experiment, which only had 6 samples, and this increases the validity of the results. So a study by Babic and Watata found that Pseudomonas was clearly the dominant microbial organism on spinach leaves, and this organism is responsible for bacterial soft rot, which can affect the fresh appearance of the spinach leaves and therefore the shelf life. Again, further biochemical testing would be needed to determine the presence of Pseudomonas, where this can be done by a gram stain and a citrate test. So the report from AH&R Environmental Proprietary Limited estimated that the plant height of the spinach will be strongly correlated to the shelf life. and they concluded that there was a decrease in shelf life for taller plant heights and an increase in shelf life for shorter plant heights. They suggest that this may be due to larger leaves being more exposed to physical damage compared to smaller leaves. So the results unexpectedly reveal that the heights have increased from all samples except one, and this may be due to error in the measurement as the shape and structure of the leaf was not flat, making it difficult to get a consistent and accurate result. So based on the results, the organic variety had a longer shelf life. So for weight, the organic samples overall had slightly higher weight loss values, and weight loss is directly related to water loss, and this is important in determining the shelf life and quality. So loss of moisture in the spinach will cause the leaves to shrivel, leading to rapid deterioration, and thus shortening the shelf life. So the experiments for Pseudomonas, plant height, and plant weight were directly affecting the shelf life, where organic has a longer shelf life in 2 out of the 3 experiments, which indicates that organic has a longer shelf life. In regards to the E. coli experiment, organic samples had more levels of contamination, which could lead to finding pathogenic E. coli in the sample, and the sensory assessment could not be made, which was important in determining the shelf life and could have changed the results. So consumers that are conscious of the shelf life of spinach can pick organic over conventional as there are lower levels of the spoilage agent Pseudomonas. And consumers that are concerned about food safety can pick conventional over organic due to the increased risk of finding pathogenic E. coli in the samples, which can lead to serious complications. This is based on the assumption that the follow-up tests have been done to confirm the presence of Pseudomonas and E. coli in the samples, and there are also many limitations to the study, which includes having restricted laboratory access and time frame of the study, and storage conditions not being met, which had a significant impact on the results obtained. So I just want to give a special thanks to the people who have helped me throughout this project. Enzo, who's been my primary supervisor, helping me with the weekly diaries and giving me feedback on what I need to improve on. Vita for getting the labs up and running and ensuring that everyone has some practical aspect that they can incorporate into their project. Callis for helping me in the labs and taking photos of the Pseudomonas samples when I couldn't attend the lab. Rebecca for ordering the suitable agar for Pseudomonas and taking out the plates of incubation when they needed to be taken out. Daniel for giving me feedback on my literature review. And Peter for giving me initial ideas to get the project up and running.